Just for the record, if there are a lot of cuts in this, I have some of the most inconsiderate neighbors on Earth and the thinnest walls outside of Japan. But at least in Japan, they know not to stomp on your head. This is ridiculous. Oh boy, alright. So, hi friends, it's Sarah again, how are you? You may notice I'm not in front of my bookcase. I broke my foot, and this is my living room. Welcome to my living room. If you hear banging, it's because I have very inconsiderate neighbors. I'm sorry. Today I'm going to talk to you about the clinical trial process, which is basically going to be a continuation of the thalidomide discussion. We left off talking about the things that came after the thalidomide scandal, and this is one of the things that was heavily developed after the thalidomide scandal. So let's talk about it. Some degree of experimental trialing has existed since, as far as we know, society began writing down its history. We have seen people in ancient civilizations doing experiments essentially to see why certain things happen. At the time, most of the experimentation was observational and it was done by trial and error. There was no real methodology to it. So in order to get to the methodology, we have to move forward. There were a certain number of trials that were sort of kind of done. They weren't necessarily called that at the time. They weren't necessarily done in the way that you would think of them now, but they maybe had a control group, or they maybe wanted to see how certain factors affected something else. So they made what would resemble certain elements of what would become a trial process. What we would know as a trial process wouldn't really come about until somewhere in the 1900s, the early 1900s, and specifically in America. We did that one. You're welcome. Prior to 1900, there were a lot of medicines going around, treatments, snake oil, call it what it is. There was a lot of things that were going around that were supposed to be helpful for curing this or doing something else, or all sorts of manners of things that these substances were going to be used to treat. Whether or not the product worked was up to the person who marketed the medication, product, and the person who took the product. And they could decide amongst themselves if it worked, if they had any positive results. There wasn't any real way to standardize that at the time, and I'm pretty sure that the snake oil salesman, literally snake oil salesman in some cases, wouldn't have wanted a process. The American Medical Association in the very, very early 1900s, the Knots, were trying to get some sort of a regulation on these products that were being used throughout the country. And at the time, the government said, we have no interest in this. So what the American Medical Association did was they created a process of approval, basically to indicate that the product was, at the very basic level, safe for a person to use. This was a very basic level, and it really equated to not much more than you're probably not going to die if you take this or have some sort of other bad reaction. These procedures were put in place to try to weed out the products that were either completely ineffective and they had no effect on whatever it is that they were trying to treat, or no effect whatsoever, or they were unpure in some way or another, or they were somehow unsafe. They would have some negative effect on the person who took them. Obviously, that's what unsafe is. The American Medical Association developed a process whereby a manufacturer, a person who wanted their product to appear in the Journal of the American Medical Association, the JAMA, they would need to have this little seal. And the seal was basically a purity seal, a seal of approval for the purity of the product. Basically that it wouldn't kill you, and it had a very basic level of efficacy. At the time, that was more or less the only way to determine whether or not a product had either safety or efficacy, and that's how it would stay for a little while. Because of the widespread use and distribution of these products in 1906, the Pure Food and Drugs Act was put into place by the government. So the Pure Food and Drugs Act was put in place to stop the interstate transportation of, adulterated or mislabeled food and drug products. This was a bare bones sort of thing, and it was at a very basic level to prevent the, again, interstate transportation of products that had bad effects, that were mislabeled, that were somehow not what they said they were. This was also the first place where we were going to see a product labeling act, which would require a label to list if the product contained any of 10 additives or ingredients that were deemed to be, what is it? That were deemed to be addictive and or dangerous, one of which being the infamous cocaine, I believe it was, opium, that sort of thing. We wouldn't see what was to be known as a trial in the way that we know it until about 1935, when there was a Supreme Court case which ruled that a person could be used as a willing subject in an experiment to gain further knowledge for the development of a product and not just for the curative process of taking whatever that product is. This opened the door for essentially human experimentation. Willing human experimentation. Around that time, it came to the government's attention that there were still a number of products that were being distributed that were in some way unsafe or not good for whoever it was that was taking them. One of this was a product called Banbar. It was essentially a sugar syrup with horsehair in it that was given to diabetics. And if you know anything about diabetes, giving somebody sugar syrup is probably not a good thing. And it was also bad because it was often taken in place of insulin, which had been developed at the time, and it led to many people getting very sick and dying. There was also a product that was used to treat streptococcus that was developed, but it was in a liquid suspension that was, roughly speaking, antifreeze. So people would take this for their streptococcus infection, their strep throat, and they would be ingesting antifreeze. These were two major scandals at the time, which led to the 1938 Food, Drug, and Cosmetics Act. Prior to the act, it was sufficient for a product distributor to have their own anecdotal evidence to say that this product is perfectly safe. It's wonderful. Banbar, for example, had a number of doctors return testimonies that, oh yeah, my patients take this, they love it, they think it's wonderful. And they used that as evidence that Banbar was safe, despite the fact that it absolutely was not appropriate for what it was being used for. With the 1938 Act, there was something called a new drug application, which was required to be submitted to the FDA if you wanted to distribute your drug, if you wanted to appropriately distribute your drug. There needed to be provided adequate evidence to show whether or not the drug was safe. It didn't necessarily specify what evidence or in which way this evidence had to have been developed or gathered. So that'll come up later. Keep that in your head. A new drug application also required that a product had information on all clinical investigations submitted. They had to submit the components and the composition of the product. They had to submit the manufacture methods of how this product was to be manufactured, in what facilities, in which way. And they had to have a copy of the product labeling submitted to the FDA in order to get approval. As of the 1938 Act, there was no agreed upon or legal or specific way that you had to do these experiments in order to gain this information that was to be submitted with your new drug application. Just prior to World War II beginning, the JAMA, the Journal of the American Medical Association, again comes in and does something outside of regulation and they publish an article which essentially laid out the appropriate way to do a clinical trial. The article would talk about experimental design, clinical trial methods, it would talk about data analysis methods, but it was specifically noted that this was not a regulation, it was simply a guide, and it was not used by everybody. But it did resemble what we would now know as a clinical trial, at least at a very basic level. After World War II, there was a great need for antibiotics, but there was also a shortage of antibiotics, which we learned from the whole thing with thalidomide development. But this would actually lead to the development of a randomized trial. There was one particular experiment where a manufacturer was trying to determine whether or not this particular antibiotic was working, but they did not have enough of the antibiotic to give it to all of the participants that they wanted to. What ended up happening instead was they would only give the medication to some of the participants in the trial, and other ones, in this case, would receive nothing because they didn't want to use a placebo at the time, something about an injection. I'm not entirely sure what that was about. But they did it randomly. And because they did it randomly, this was able to eliminate what was a relatively well-known bias at the time, where a number of people who were doing trials would give the trial medication to their more healthy patients and not give a treatment or give the lesser treatment to their sicker patients. And Obviously, the sicker patients would generally have poor outcomes, and the more healthy patients, regardless of whether or not the medication worked, would have better outcomes. And it would obviously give a little more credence to, oh, well, I guess this works. Whether or not it actually did, it didn't make a difference. It was a bias. But because of this one particular experiment, they were able to figure out that they could eliminate that bias, which is fabulous, right? In 1948, the results of the trial were published in a journal, and the people who did the experiments were praised for bringing in a new era of medical experimentation, or new era of medicine anyway. So we suddenly have a randomized trial. Excellent. But because of the fact that they had discovered this new way to standardize a trial, this would lead to further developments in other ways to standardize a trial or to have a better way to gather results from a trial. Developing from the idea of a randomized trial, a double-blind studies were preferred because neither the doctor nor the patient would have the bias of knowing who was receiving the medication or the treatment, and it would eliminate the bias of saying that I've taken this and I feel better, or I've given this medication and she seems better, something to that effect. It would also standardize some more of the methodologies for data collection, for distribution of medication, for the ways that medications were taken. It would allow for certain things such as a uniform schedule of distribution. It would allow for the uniform taking of data. It would allow for standardization of observations. It would allow for a number of very good methodologies that lead to very good science. Of course, after World War II, there was a lot of question about the efficacy of having a placebo-controlled study where one group would be receiving a treatment and the other would not be receiving a treatment for an illness, which potentially was doing them harm. But ultimately, it was decided that it was the best way to get good results, and out of all of this great experimentation came the gold standard of a clinical trial, which is to say a randomized, placebo-controlled, double-blind study. So a randomized, you don't know who is and who isn't getting the drug. Double-blind, the patient nor the doctor know who's getting the drug. And placebo-controlled, meaning that the effects are seen of just the drug and nothing else, in theory. But like I said, the 1938 Act was more of a suggestion in terms of testing instead of a regulation. And in 1958, what's his name? Senator Estes Keffer had a trial put in place to address the fact that there was so much inconsistency with the information that was being submitted by these medication companies, drug companies, pharmaceutical companies, manufacturers at the time in order to get approval for their products. It was noted that an awful lot of these products would completely skip animal trials and go straight to human testing. And this would lead to a number of instances where the results were that they did not know that an effect was going to be seen until after it was seen in humans, which could have been caught earlier. It was not a good place to be in terms of experimentation. One of the ways that information was gathered, if they weren't doing a trial, which many of them weren't, was that the company would hand out samples to doctors, and they would say to the doctor, tell me how your patients like this. And the patients would come back and say, yeah, it worked, or it didn't work. And the doctors come back to the pharmaceutical company and say, well, it's good or it isn't good. That seems to be what more or less applied to the study that was done by Smith, Kline, and French when they were seeing whether or not thalidomide was going to be good for distribution in the United States. That's basically how thalidomide made its way into the United States for what little did. It was specifically noted that the patients who had been giving this experimental drug had no idea that they were taking an experimental drug, and they were essentially unwitting experimental subjects. They didn't necessarily want to be an experimental subject, and they had no way of knowing that they had been experimented on. You'll note that it was 1958 when this trial had begun, and this kind of bounced back and forth for a while. The manufacturers of these products, they came back and said, well, it's very expensive to hold human trials. We have to throw out all sorts of medications, all sorts of products that don't work in order to get the one that does, and then we have to pass that on to other to our consumers, and it's just not economical. And they had a whole laundry list of stuff that they had to say against the idea of having any sort of regulation on this. Of course, the thalidomide scandal happened, and almost immediately it was seen as necessary to have the regulations put into place. In 1962, our good old friend Francis Ullman Kelsey came out and said this: in no time, the fighting over the new drug laws that had been going on for 5 or 6 years suddenly melted away, and the 1962 amendments were passed almost immediately and unanimously. So in 1962, the Keffer-Harris Act was passed to place a number of different regulations, and the next year there was the 1963 investigational drug regulations that were put into place in addition to that. One of the most important things to come out of the 1962 regulations was that there was pre-trial evidence required to say that this product was safe to use in trials, which meant essentially that there had to have been animal trials or some other related trial to prove that this was not going to be harmful to a human patient, which, if you remember correctly from the thalidomide scandal, was not done in that case. In order to get approval to test your product in a clinical trial, you would have to apply for a new thing, which was called an investigational new drug application, an IND, which precedes the new drug application, which came out of the 1938 Act. The IND would supply an alert to the FDA that a manufacturer was going forward with a process for this. It would provide animal toxicity data to prove that it would be safe to move on to human trials. You would provide manufacturing and processing info so that the FDA would know how this product was going to be manufactured. It would provide the chemical background of the product so that you would know what was in it. It would provide a layout of the clinical trial with safety measures so that the FDA would know who was doing it, what were the qualifications, the facilities in which these tests would be performed, and the populations in which the tests would be performed. All of this would require an institutional review board approval of study. So the review board would say that it is okay to do this study. I've seen your layout of how you're going to do it, and we approve of this study. Every single person that was involved in the trial would have to sign off and say that they were going to conduct the trial as it was approved by the review board, that they would personally conduct and supervise the test, that they would inform the subjects that the drug is for investigational purposes, and that they would report adverse events to the sponsor of the study. So if anything went wrong, they had to report it. Think about that. One of the biggest things to come out of the 1958 Act was the idea of efficacy, which is to say, whether or not this thing works. We saw a very, very small bit of that in the very, very early days, essentially separating snake oil from some kind of treatment. But if you remember correctly from the thalidomide scandal, thalidomide didn't appear to have any particular sedative effect, and because of that, the people who took this, the mothers who took this medication and had this great effect on their babies, they took it for no good reason, it seems. The 1958 Act required the manufacturers to prove that this medication, this product, has the effect that it purports to have in the way that it purports to have it. This all had to be done before the product could go into the marketing phase. In the original thalidomide video, I did say that the people who were doing the studies had to prove that the medication had the effect that it was meant to have without a reasonable doubt. That's not exactly correct. It's more that is the ideal situation, but more important is that there is substantial evidence that the drug will have the effect it purports to have or is represented to have under the conditions of use prescribed, recommended, or suggested in the proposed labeling, which in theory is a preponderance of evidence beyond a reasonable doubt, but the Congress at the time was concerned that if that was in place, that it would be too high of a standard. The law states that the information has to be from adequate or well-controlled studies, but it specifically does not define what those terms mean. In addition to all of that, the 1958 Act would require that all of the medications that have been given approval since 1938 and even before had to be reviewed for their efficacy. And thousands of medications would come in, a little bit over 1,000, I think it was. And what they would find was an incredible number of those studies were unbelievably inadequate. Some of them skipping human trials, as we mentioned, or skipping animal trials, as we mentioned. Some of them were ridiculously useless. And we saw that with the thalidomide studies, or the fact that they tried to put it through with no studies at all. Clearly this was going on. Let's talk about phases now. We are generally aware of the fact that there are 4 phases to any trial. We talk about the phases, the pre-trial phases, where you're going into animal studies. You're talking about information that is gathered in a laboratory to lead into whether or not you can do a human trial. Once you get into the human trials, you have 4 phases, phase 1, 2, 3, 4. We're going to imagine that we have 1,000 applications for new products as we're going through this process. Phase 1. Phase 1 is usually from a few months to a year, anywhere from 20 to 80 participants. Generally, phase 1 studies are done on healthy participants, and it is to gather information on whether or not this product is going to be safe for humans and determine at a very basic level where dosing should be. So what they do at this level is that they begin to give a very, very low dose to the subjects, and if that's good and it doesn't appear to have any negative effects, and maybe it does something, they go to a little higher dose and they tweak that until they have something that is good enough to go on to phase 2. During phase 1, they also work on the best way to administer the medication. So are you going to take it in a liquid suspension? Is it going to be injected? Are you going to get a pill? Are you going to get a suppository? Is it going to be a cream? What have you. Approximately 70% of applications will move on to phase 2 from phase 1, which if you go from our original 1,000 applications, about 700 of those are going to move on to phase 2. Phase 2. Phase 2 starts with anywhere from 100 to maybe a few hundred participants. You don't generally see it going above a few hundred participants, and this is the first place where they're looking to see if the medication, if the product is effective. This usually lasts anywhere from a few months to 2 years. The reason that there are so few participants is because phase 2 is closely monitored and well controlled to determine whether or not this product is going to be effective. This is also the point at which they may try to find combinations of products and see what the effects are going to be on that. So phase 2 is going to look for the treatment of symptoms, or it could also possibly look for surrogate outcomes. So instead of specifically treating maybe what it's meant to treat, it could have an effect on something else, which can have an effect on the original problem. So one of these things, for example, is there are a number of products that have an effect on lowering blood pressure, despite the fact that that's not what they're meant to do. Ideally, at this stage, you're looking for the gold standard of testing, which means that you have the randomized, double-blind, placebo-controlled studies. Sometimes during this phase 2, they will also allow for the comparison of the new treatment to what is the standard treatment for whatever is being studied to. This is also the first point at which they consider side effects of whatever it is being tested. Specifically, in this stage, you're looking at patients who do have the condition being studied. So they are seeing whether or not it works. They're seeing if there are side effects, in particular in relation to whatever the condition is, and also in general. The sample size is not big enough at this point to determine what the generalized risk is going to be, but it is used to set up a bigger experiment for phase 3 to determine what the generalized risks and benefits are going to be. Approximately 33% of applications move on to phase 3 trials, which means that an awful lot of them fail. So if we're looking at our original 700 applications that moved on from phase 1, we're now down to 231 applications that will move on to phase 3. Phase 3 is generally a few years long at least. You're looking at a study that involves hundreds to possibly thousands of patients. This is the point where they've proven that the product is effective for what it's trying to do, and here's where they try to determine if it is more effective than the currently available treatment, whatever that is. They do this by determining whether or not this treatment has a greater reduction of symptoms and or fewer side effects than the currently available treatment. They gather this information from this much wider population so that they can determine what the side effects are going to be. They can determine better dosing. They can determine if there are going to be different effects based on different populations. This is where they get their risk-benefit determination. This is where they decide if the risk of this new medication is worth the benefit that is going to be derived from taking or instituting this new treatment plan. The way that they are able to determine the cost-benefit, the risk-benefit analysis, is by the quantity of subjects that they are studying. So when they have a lot of people over a relatively long period of time, they're able to gather a lot of information about whether or not this treatment is going to be safe over a wider population and whether it is effective over a wider population. So ideally, it's a double-blind assignment of who is going to receive the treatment versus the old treatment or whatever the other option is. The product is tested among a wide population. It's tested to see what side effects are going to come up. It's tested to tweak the dosing information based on those populations. For example, small women versus large men, possible ethnic differences, that sort of thing. This is also the point where the preparation of the labeling information, which is very important to move forward, begins. If this product is given the okay, it moves forward to get its NDA, its new drug application, from the FDA. From here, the FDA is going to decide if this is safe to move forward. It's going to determine who's going to take this in what dosage based on the information that was provided by the trials. From this point, we move on to stage 4 trials. Approximately 25 to 30% of the products that make it to stage 3 will make it onto stage 4. If we're talking about our original 1,000 applications, from that moving on to stage 4, there are between 58 and 69 applications that will move on to stage 4. The FDA, of course, has the opportunity to request more trials based on the risk-benefit analysis, based on any other information that's provided in the application. There's a caveat to all of this because of a 1992 regulation that was put into place. It was sort of a addendum due to what was happening during 1992, the AIDS epidemic, and a few other, primarily the AIDS epidemic. There was a big push for medications that may be in early stages of trials to be given to people who were very, very sick. In the 1992 regulation, it was determined that based on the results of those surrogate endpoints, the things that may have indicated some sort of an effect, whether or not the final effect was seen, the regulations allowed for medications that had positive surrogate effects to be used on the people who may have needed to have some sort of a treatment because of a serious illness at the time. In 2012, there was an amendment to the Food, Drug, and Cosmetics Act based on whether there was an illness that had an unmet medical need. This would allow for a medication that did not have full approval to be used in a therapeutic way. So, for example, if somebody had a serious cancer, and there was a surrogate effect to whatever this medication was that's being studied, where a tumor would shrink. Now, maybe the target effect was a longer lifespan or complete obliteration of that cancer. The fact that the tumor shrunk would have been an indication that this medication had some kind of an effect. Because of these things, they may have been able to apply for accelerated approval. And this wouldn't necessarily approve the medication. It would simply allow for the distribution of the medication to patients who may have had a high need for the medication without having fully met the standards of clinical trials that are otherwise required to prove that the medication, that the product is safe and effective. When you're giving out a product that isn't fully approved, you're going to expect that that will be under further testing, and that when the results of those tests come in, this approval can be revoked at any time. It can be modified at any time, and it does not mean that the medication, the product, is fully approved. It simply means that it has an approval for use. Specifically, Phase 4 trials, which we're going to talk about in a minute, are crucial to whether or not the approval will remain intact. Phase 4 is simply post-marketing review. So sometimes what will happen is a manufacturer will study a group of people who are currently taking the product or using the product, and they will do further studies on a greater population about, again, safety and efficacy. there have been a number of products that have been recalled over the long period of time that we have had these regulations in place. And they've been recalled for any number of reasons, ranging from neurological conditions to heart conditions. Often what's seen is liver and kidney inclusion in bad outcomes. Occasionally, things such as alopecia, which is the loss of your hair, or it's ichthyosis, which is a very serious skin condition. Things like that may or may not be seen. Sometimes psychological conditions can arise out of taking certain medications. A number of medications that have been recalled have actually been related to weight loss, which is interesting. I'm going to link a listing. I don't particularly like Wikipedia, but it is a good place to gather information. They have a listing of recalled information that runs from somewhere in the 60s to current, which I actually found a medication that I used to take on there occasionally, which was recalled 2 years ago. So I'm glad I looked at that. Phase 4 also allows for the removal of drugs which have addictive or overdose problems that are related to it, or it also allows for the removal of medications, products that somehow have gotten through the rest of the process that simply aren't effective or as effective as another treatment, for example. If you've ever seen a commercial for a medication, for a pharmaceutical, that says, if you have trouble affording your medications, please call yada yada yada, and we may be able to get your medications at no cost. Or there was a medication that I was prescribed recently where they had a helpline where you could get your medication for free, presumably for being a part of the stage 4 studies. What is the final point of going on that entirely too long tirade about clinical trials? By the way, I'm not a scientist. I am just interested, and I think this is relevant to a lot of things, including what I had previously talked about, which is the thalidomide scandal, and possibly other scandals that have arisen since, before, or around it. I think it is important that you know the process through which most things go. I think it's important that you know how things come to be so that you can make decisions based on knowing what normally happens. If something abnormal happens, you have the information to know that that is abnormal, and that process is something that you need to take into consideration when you're making a decision about if you want to do something or not. You need to have all of the information that you can in order to make good decisions. And this is simply one other way that you now have good information to make good decisions. If you're paying enough attention, you may be able to figure out what this is in relation to, but I'm probably not going to say it because there are certain things that you're not allowed to say on the internet right now. I think it's very important that consumers know the normal process that things go through so that they are able to determine when things are unusual. One thing that you're going to be able to do because of this is you're going to be able to do your own cost-benefit analysis. You are going to be able to determine for yourself, in many circumstances, if you think something that you want to do, need to do, is worth the risk, if the benefits are worth the risk. You have choices in these situations unless you're being coerced, right? So take the information that you're given and make good choices. Determine your risks, determine your benefits, determine the costs that are associated with anything that you do, and know that you are in charge of your own self and your own life. Advocate for yourself. Use your head. Have a good day. I'll see you next time. I'm hoping that some of the next talks that I do are going to be a little less data heavy and talk heavy and a little more philosophical maybe. Thanks. I'll see you next time. In 1962, Frances Oldman Kelly, our good old friend Frances Oldman Kelly, Kelsey, her name is Kelsey.